Today we begin section 4, which is about antibodies and B-cell development, and this lecture will look at antibodies and the various structural elements of the antibody. Antibodies are divided into a series of domains, and we have two chains, types of chain, a heavy chain and a light chain. And here's one heavy chain, here's another heavy chain, and here's one light chain, and here's another light chain. And these chains are divided up into domains. The light chains have a variable and a constant domain. So C and V stand for constant and a variable. L stands for light. As for the heavy chains, they have just one variable domain and 1, 2, 3 constant domains. There are a few antibody types which have more than which have a fourth constant domain as well. Each of these domains basically comprise a specific type of pattern, structural pattern called the immunoglobulin fold, which is basically a 3-stranded beta sheet folded over a 4-stranded beta sheet represented in red and blue respectively. And this yellow bar joining the two is a representation of a disulfide linkage between two cysteine molecules in here. So this is a stable structure, and this immunoglobulin fold is the basis of all of these domains. Some variability happens in the exact structure and location of these loops, but this folding pattern is conserved amongst the antibodies. Amongst the domains in antibodies. In the next slide, we'll look at this structure in a bit more detail, but I'll just add one more thing here. These little lines that represent a disulfide linkage which holds together the two heavy chains. So the heavy and light chain are held together by weak non-covalent interactions, and also the constant domains in this region here are held together by non-covalent interactions, but disulfide linkages hold together the two heavy chains, forming this structure. Now, coming over to a slightly more detailed look, here we have light chains, variable, constant, heavy chain, variable, constant 1, constant 2, and constant 3. Here are the disulfide linkages that hold these two together. And experimentally, people have named various parts of the structure of the antibody based on enzymatic cleavages. And it's possible through papain and pepsin to cleave the two heavy chains either on this side of the disulfide bond or on that side of the disulfide bond. If we cleave on this side of the disulfide bond, we end up with two types of fragments that can be separated electrophoretically. So if we cleaved here with papain, then we'll end up with this. The disulfide bonds are holding these together. And this fragment is called the FC fragment. And C stands for crystallizable because it's all constant here. There's no variation amongst antibodies. And something which is a pure repeating unit can be crystallized. So this whole term is something purely experimental. But it links to the function because the fact that this is constant is important. Once we've cleaved with papain, we end up with two FAB fragments, and F stands for fragment, AB stands for antibody binding, and the antibody binding is mediated through the variable region here. If we were to cleave with pepsin, we'd end up with this is one unit because the disulfide bonds here would be holding everything together. So we'd call this an FAB prime 2 fragment. This terminology is not going to be very practical for you, but the terminology that is very important for medical clinical applications is FAB, fragment of antibody binding, because that contains the variable domains, and the FC fragment, because this is where complement factor from the classical pathway binds to this region, and also something called an FC receptor is able to bind to this region, and that's physiologically and medically extremely important. Some smaller points that probably aren't very relevant to you at this stage in your education are that there are actually a number of intra-chain disulfide linkages. So here, 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 down here, down here, down here, and so on. But this disulfide, which is interchain between the two chains, that's critical to understanding the structure of the antibody. And finally, this CHO here, this refers to glycosylation, and antibodies are in their constant domains glycosylated in various different places, and this changes depending on the antibody class or isotype. So you heard of IgG, IgM, IgA, IgD, IgE. They differ in the locations and places of glycosylation. Now, looking just at the variable domain, and here we have the light chain variable domain and the heavy chain variable domain. And if we look at these loops here, which are, some of them are colored, then there's a certain interesting property here. So the speed immunoglobulin fold of two beta sheets folded over each other make the core of the domain. However, the loops between the beta strands that make up the beta sheets, these all protrude outwards, and this is surface exposed. If we were to ask ourselves, where is the rest of the antibody? Okay, so the constant region would be here, and then the constant region of the heavy chain would be here, and then the heavy chain would continue going down the slide. So we've removed all the constant domains here, and we're just looking at the variable domain. And this area here is what will be surface exposed and able to recognize antigens. And people have looked at the sequence variation in variable domains across the whole length of the domain. So looking across all the amino acids one by one, and for each position, noting how much variation you have. And what you'll find is that we have peaks and troughs, and these peaks up here, here and here, represent specific loops, 1, 2, and 1, 2, and 3. And the same for the heavy chain, H1, 2, 3, are represented here as distinct peaks of very extensive variation. Huge difference between the amount of variation up here compared with down here between heavy 1 and heavy 2. And we refer to these as complementarity determining regions, or CDRs. So if you see that people talking about CDRs, complementarity determining regions, these are those regions which are very variable. And the word complementarity is referring to the fact that these are complementary, or able to bind to antigens. So the ability of our antibodies to be able to bind to a whole range of different antigens comes from this variability here. So we can select thousands of B cells and sequence, let's say, thousands of mature B cells, and sequence the variable region, and we find all these differences which are focused in these points, and that gives B cells a huge range of potential binding sites. The molecular mechanism which generates all this variation is called, well, it's a somatic recombination event, and we'll talk about that not today, but in a following lecture. So we looked at the variable bit. Now let's look at the FC, which is the crystallizable bit. And here we have two domains, one in immunoglobulin domain, another one. These are, so we've got two heavy chains attached to each other, here and here. And the people have mapped various different regions here. So this bit in red here refers to the place where the FC receptor, and in this example for FC gamma, so this is a FC receptor that binds to IgG, recognizes this part of the FC fragment. Complement C1q, which is from the classical pathway, and that recognizes antibody, which has been bound onto some pathogen, and that is mediated through this region here. Remember, all this is constant, so that means that complement C1q can recognize any antibody. In this case, then, any antibody from this class. And here is something called the neonatal FC receptor, for FcRn for short, and that is recognizing here. So this is the antibody, and when I say that these colored regions are the regions where these various receptors or complement factor can bind to each other. And I mentioned about the classes. Here we've got an IgG, but let's look at the various different classes on this table. We have IgG, A, M, E, and D. Remember that if we refer to the polypeptide or the gene, then we use the Greek letters. So instead of G, gamma, alpha, mu, epsilon, and delta. And that comes in when we look at the individual polypeptides, so gamma, alpha, mu, epsilon, delta. The light chains are, in terms of their constant region, are the same for all of these different classes. So there's no Greek letters here, so it's just the light chain. And there, of course, there are two of them and two heavy chains. And we can have something called subclasses. So we have IgG1, IgG2, IgG3, and IgG4. IgG is the monomeric, and we can have these various different classes. And these are actually derived from unique constant regions within the FC fragment. The light chains stay the same in all of these classes, but the heavy chain varies. Where are these found? Serum, primarily serum. IgA is in serum, and it's also secreted, and so this is considered to be a mucosal antibody, and these also are found in serum. Information about complement activation. Not all of these are able to be recognized by complement. IgA can also have two subclasses, IgA1 and IgA2. Some of these, like IgA, exist as monomers, and IgA can also dimerize with the addition of what's called the joining segment. J stands for joining. And in the process of secretion, then they also have something called a secretory segment. And actually, we will look at this process of IgA secretion later and the physiological relevance of that. IgM is pentameric, and the two heavy, two light chains, you also have a joining segment. You'll also see the word isotype. So sometimes the word class and isotype are used interchangeably. So when we talk about class or isotype, then we're meaning the same thing. This is IgD class or IgD isotype. Structurally speaking, we can see a commonality between these different classes or isotypes, constant domains, variable domains, heavy and light. But when we look a bit more closely, we can see some subtle differences. So comparing IgG1 and IgA1, for example, the hinge region of IgA1 is much longer, much more extended, and IgA has some kind of a tail here, which doesn't exist here. These little squiggles here represent the sugar residues which are attached at different positions. So they also differ in that respect as well. IgM actually has a fourth constant domain in its heavy chain. So IgG is 3, IgA is 3, but IgM has a fourth. IgE also has an extra constant domain. Much more glycosylation on IgE. IgD has a huge hinge region with some distinct regions of positive and negative charge on the amino acids, forming some loops here. Also a tailpiece like IgA. Remember, IgA can be secretory, and that means that there's this secretory component in light blue which holds these two together, or actually what holds these two dimeric forms together is this J chain, which is an extra chain. And in order to be able to be secreted as a mucosal antibody, then it needs to have this secretory component also present. IgM, there's something more to say about IgM, isn't there? Because the last slide said that IgM can be pentameric. So IgM can be joined by a J chain, which joins together 5 of these individual units into a pentameric structure, or star-like structure. And here is actually an electron micrograph of such a structure. This molecule represented is planar, meaning just that it's flat. Sometimes we can actually have a few IgM units that form a hexamer, but it's not clear how relevant that is physiologically. The pentamer in the planar form is like this. When it actually engages antigen, then it forms this crab-like structure, and the complement recognizes IgM in this conformation as opposed to this conformation, which is a good thing because that's a mechanism for ensuring that complement gets activated only when IgM is actually bound to to antigens. We don't want complement just triggering itself randomly as it encounters antibodies in our blood. So complement and antibodies are floating around in our blood, and we don't need these to react with each other. However, upon binding, some small conformational changes occur, and this is, for the case of IgM, is recognized quite easily by this change in the way the arms folded. And you can actually look at that. This is an electron micrograph, and here you can see the arms are folded in as opposed to this planar flat structure over here. Here are a list of some subclasses, but this figure is taken from a paper titled IgG Subclasses and Allotypes from Structure to Effector Functions, and I just want to introduce a few small concepts here, just two concepts actually. And the first of these is the concept of the allotype. And allo is sounding similar to allele, and an allotype represents a polymorphism within some constant domain. So within the population, then the IgG1, for example, of my antibodies might be different from the IgG1 of your antibodies. There are actually not that many allotypes that have been identified. There are a few dozen allotypes have been identified for IgG, for example, in the heavy chain, and a smaller number in some of the light chains. So that's allotype. Why is allotype important? Well, it can be very significant medically when we want to use antibody therapy. Antibody therapy is very important in treating some cancers, and it was also considered helpful in treating acute cases of COVID. People given antibody treatment. Donald Trump got antibody treatment when he had COVID. Monoclonal antibodies. And in treating cancers as well, there are various therapeutics which are monoclonal based. And yeah, so the monoclonal antibodies are important as a medical intervention. The problem then is that we may have an allotype which doesn't match with the monoclonal antibody which is being produced. The word monoclonal means that it's simply a very specific single type of FV. In our blood, after you have an infection, there'll be a huge number of different antibodies with different variations in their complementarity determining regions, all of which are capable of binding to the pathogen, but they're binding in different places and in different ways based on their difference. So that's a polyclonal antibody. So if we take blood and separate the serum, then this serum will be polyclonal, and people have used that as well actually as treatment. Someone who recovered from infection, take their blood, some of their blood, and separate the serum. That will be full of a big mixture of antibodies, most of which will be directed towards the very recent infection, and that may be used to cure another critically infected person. But that's polyclonal. In terms of a medical intervention, more commonly, more commonly pharmaceutical companies have invested in isolating or engineering very specific antibodies, and these are monoclonal antibodies. But because of this small allelic variation that can occur in the constant regions, there could be a mismatch. So this is where allotyping can be important. As a subcategory of this discussion of allotyping, sometimes an allotype actually is allelically different, say for example from IgG. However, that allotype may be the same as in IgG2. In other words, just supposing that at any particular position we have, say, an alanine in this chain here, and in IgG2 we normally have threonine. But there may be in the population some rare cases where people have threonine here, so that's an allotype because it's a polymorphism, it's a mutation. However, threonine is normally found at that position in IgG2, or rather, if in this hypothetical example threonine is found in IgG2, then actually that means that at that position it's similar to IgG2, so it's called an isoallotype. And that's important because in that case that threonine won't be immunologically important because your body's already been exposed to that sequence present within that constant region. Also, some of these allotypes may refer to sequence mutation in a place which is buried deep within the structure and is not exposed to the surface. So even though there's a polymorphism here, and we are receiving monoclonal antibody treatment with the other form, but immunologically that's not significant, and the monoclonal will still be recognized as self, and we don't have self antibodies. If we did, we'd be dead, because we wouldn't be able to fight any infection, because we would have our own antibodies attacking ourselves. This is also from the paper. This is relevant for genetic engineering of antibodies. And just an interesting thing here, the concept that people have developed engineered antibodies where one FAB fragment is different from the other. So you can see there's some color differences here. And this color difference, as opposed to hair, for example, is supposed to represent the idea that this FAB fragment is recognizing something different to this FAB fragment. And the significance of that is that you can then cross-link two antigens or even two cells together. And an example of two cells is we may be looking to try and cross-link a cancer neoantigen from a tumor cell with a complementarity determining region on a T cell. So the T cell receptor, very similar to the the FAB fragment variable domains, also has complementarity determining regions, and we'll look at that in the section that follows this one. So this way you can actually cross-link two cells. So this is an area of research, this technology of making bispecific and antibodies as a therapeutic. It's something people are interested in. Also, it's possible to alter the disulfide bonding so that the cysteines which form disulfide bondings here may actually be replaced with an intra-chain disulfide bond. And this alteration between these intra and interchain, these are called isomers. This part is not very relevant for a medical application, but the allotype section is, and you may come across bispecific, the engineered antibodies if you read research about novel treatments. Now that we have defined isotype and allotype, we need to look at ideotype. Ideotypes are important in drug discovery and design. Let's set up the definition of ideotype, and then we'll be able to understand what an anti-idiotypic antibody is, and these are what are important in the process of developing and determining the pharmacological behavior of therapeutic antibodies. It all focuses around the variable domain. First of all, when we consider the interaction between the variable domain of an antibody and the antigen, where the antigen could be a whole bacterium or a specific protein, within the antigen there will be a very specific region which is actually recognized like a key fitting into the lock, if you like, similar type of terminology used for enzyme substrate interaction, a shape-specific interaction. That part of the antigen which is actually seen by the variable domain is called the epitope. The part of the variable domain which sees the epitope is called the paratope. So you have paratope and epitope. Ideotype refers to regions on the variable domain close to or within the antigen or epitope binding domain. So these areas here, this is the paratope, and those specific regions here are also called ideotopes. But ideotopes don't have to be within the paratope, they can also be nearby. So here in this particular slide, these are labeled as ideotopes. And this whole region, which is unique from one antibody to another within within an individual, if we look at you or me, then our IgG1 is all going to be the same in the constant domains. However, we have a whole range of different ideotypes referring to the fact that we have IgG1, for example, which is some recognizing a recent COVID-19 infection, some recognizing recent rhinovirus infections and strep throat, and you know the list can continue. So we'll have a big mixture of IgG1, each recognizing something different. And this whole region is called the ideotype. And you can make antibodies experimentally in the laboratory. You can make antibodies which bind to the ideotype. And the reason that these are useful is that if you want to get approval for a new antibody therapy against a cancer, for example, then you have to demonstrate an understanding of the pharmacokinetics and pharmacodynamics of this drug. So the monoclonal antibody that we're talking about is a drug to target some pathogen or a neoepitope on a cancer cell. We call it a neoepitope because it's something it's a self-epitope from the cell, but it's something new which hasn't been recognized during the central tolerance or peripheral tolerance. So it's something which can distinguish that cancer cell from the normal cell within your body. So those cancer epitopes, those are called neoepitopes. So, I mean, that's just an example of why we may want to use antibodies as a therapeutic, of which there are so many. Now, to understand the pharmacokinetics and pharmacodynamics, amongst all the all the antibodies that we have in our body, we want to be able to understand where the therapeutic antibody is going, which tissues is this antibody being found in, and over time. If we talk about time, then that means we're talking about pharmacokinetics. If we're talking about place, that means primarily that we're talking about pharmacodynamics. If we want to understand that, then we need antibodies that can recognize these ideotypes so that we can distinguish our therapeutic antibody, which we're studying, from all the other antibodies that are present, which are all going to be the same, or maybe the same in this respect, assuming that we have a good allotype matching. So the drug and our own antibody should be the same here in this region here. We want to have allotype matching. So how can we distinguish the therapeutic antibody from our own natural antibodies? Via the ideotype, because of all this variability here in these in the three complementarity determining regions of the VH and the three CDRs of the VL domain. And in order to do that, we need to make anti-idiotypic antibodies, and then we can use it as a tool in order to see what's happening, to measure over time the disappearance of our therapeutic antibody, and to measure the distribution of this antibody in our body. The anti-idiotypic antibodies may be recognizing specific idiotopes within the paratope, in which case that antibody, the anti-idiotypic antibody, is actually able to measure free antibody, because if the epitope was bound, then those the paratope is no longer visible to the anti-idiotypic antibody, which is recognizing idiotopes within the paratope. Alternatively, the anti-idiotypic antibody may recognize variable features outside of the paratope, in which case the anti-idiotypic antibody may be able to recognize both bound, epitope bound, and unbound antibody. In other words, total antibody. So we could have a panel of anti-idiotypic antibodies, which are tools to use whenever we're interested in measuring total antibody present in the body and we want to measure free antibody. More rarely, you can even have anti-idiotypic antibodies that are actually recognizing the paratope-epitope combination. So in that case, they're only recognizing bound antibodies. So it's possible we're using a panel of anti-idiotypic antibodies to recognize bound, free, or total therapeutic antibody within the body.